In this section, we're going to talk about bacteria and archaea. So these are organisms many people are familiar with. Mainly when we think about bacteria, we're going to be thinking about bad bacteria. But they are the oldest, simplest, most numerous organisms on the planet. One of the first organisms to even be arriving on the planet. They are all over your skin, all inside your body. You have more bacterial cells in and on you than what you have human cells. I sometimes think about this when I'm like late at night and reaching for a bar of chocolate, that it's not me, it's my bacteria that are controlling my body because they need some sugar and some cocoa, all right? But they're very important in terms of what's happening in soil. They are what suppresses diseases. They are what's speeding up decomposition. They hold on to nutrients in their body, and they are what form the very fine soil microaggregates. So yes, they're very important. They do breed up very fast, and they're absolutely critical. However, when we start to see bacterial dominance in a soil, it can create certain issues. So when we get a big buildup of bacteria, what they cause is compaction layers. We can see surface crusting starting to form. If we get these very fine microaggregates, they'll move down through that soil profile, and they may in fact form hard pans. When we see bacterial dominance, that's when you're going to see the increased need for inputs and costs. One way to think about these bacteria and archaea, so archaea literally, it means the ancient ones. They are the most primitive organisms on the planet, and those primitive soil conditions are what lead to the germination signals for many of the weeds that plague us in general, and I think of weeds really as indicator species. Many common weeds that we see in grassland and in parks are really those species that are our bacterial dominated weed indicators. As we see more and more bacterial biomass in a soil, it also leads to what we call a germination signal. So these microbes are releasing all sorts of metabolites all the time, all sorts of signals, and they will signal to different specific plants to say, hey, I think you should germinate. These are the perfect conditions for you. So what's driving that germination of different species that may or may not be helpful for your enterprise is based on the biological component in your soil. Ask the question, what is it that is creating these highly bacterial soils? So bacteria themselves like to feed on very, very simple carbons or simple sugars in the soil. They also like what we call the disturbance event. So we've talked about what disturbances are. So things like our pesticides, our fungicides, nitrogen fertilizers, high soluble fertilizer rates, compaction, all of this actually stimulates those bacteria in that soil environment. And so when I think of many park spaces or like looking at New Zealand dairy industry, what we're putting on is, nitrogen fertilizers. We're using a lot of fungicides and pesticides. And what that starts to create is more and more bacterial soils. And the more bacterial that we get, then the more pests, weeds, and diseases we start to create. And we end up on a treadmill of increasing inputs, decreasing outcomes for the long run. When we're thinking about these bacterial soils, we need to be asking the question of what's creating them, and then how do I turn that around? We talked about bacteria. Now the next component is the fungi. What are fungi doing in our parks? Well, they're very important for disease suppression. They are what will actually outcompete. They will consume many pathogens. They wrap themselves around plant roots and protect plants. They are also retaining and then helping to release nutrients. So their bodies can actually help to move through these threads, and hyphae will move through the soil and move nutrients around. They are also really important for decomposing stuff. We see fungi have a direct relationship with plant growth. So as we see fungal biomass improve, we also see plant biomass improve. They are a critical component in our water use efficiencies. There's some very interesting studies coming out about the relationship between fungi and water. So get really interested with more and more pressure coming under park managers in terms of water security. Fungi play a key role in this. One thing that fungi do is they hold soils together. And soils really is our greatest resource and our greatest export. We continue to export more soil than the soil that we're building. And, we can look at some images from the 1930s from America and Australia and just see the extraordinary amount of soil that's being lost. But still, we continue to lose soil to this day. For every ton of grain, for instance, produced, we're losing 1 to 2 tons of topsoil. Anywhere between 4 to 50,000 pounds an acre a year of topsoil is being lost in America. And it's going into our waterways or up into the atmosphere. So you would have seen dust storms, you see water being discolored by the presence of soil. So in that soil that's moving into waterways, it's also carrying nutrients, it's carrying pesticides, herbicides, fungicides, and sediment into our waterways. So there's a very simple test that we can do for this, which is our slaking test. It's one indicator for air and water movement, an indicator for microbial health. So those microbes release different types of substances that will stick and hold those soils together and stabilize them. So we also call this slaking test the aggregate stability test. It's absolutely critical for resilience and the resistance in that soil so we can deal with a disturbance event. So that might be flash flooding, or we can deal with extreme heat or cold, and those soils can recover really, really quickly. So the slaking test is one way we can indicate what's the stability of those soil aggregates, what's the erosion potential, and it's an indicator for all these glues and microbial byproducts. And you might think when we're managing parks that we're not losing soil, but actually, in fact, you might be. A ton of soil is the equivalent of the thickness of a sheet of paper, and we are literally dying by 1,000 cuts with each ton of soil that's being lost from the environment. And so with a slaking test, you can tell, is that water moving through the profile and then out into waterways? So the image here shows soils that were taken about 10 feet apart. The one on the very left was inside a greenhouse. They were having some real issues with the health of the particular crops that they were growing. And you can see that because there's no stability in the soil, there's very low biological activity, it will be very bacterial dominated, that the water rushes into those pore spaces, those pores collapse, and that water becomes discolored. The vial right next to it is from soil that was taken outside of the greenhouse where you have good biological activity, and notice that that water is running clear. That's what we want to see for all of our ecosystems right across the planet. This, for me, is probably one of the best fence line images, comparing what happens when we build soil to what happens when we don't build soil. This image was taken by Kim Deans in Tinga in New South Wales. They experienced some pretty big fires in 2019. That fire ran through these properties. Luckily, they've been practicing adaptive management for a very long time. They had been building soil, they were using biodynamics and composts and compost teas and extracts. And what you can see is along the fence line, the neighboring property behind them is blowing. That dust is coming up off that ground, but yet when the wind moves across their property, no soil is moving. Right? Australia lost hundreds of thousands of tons of topsoil in 2019 and 2020 because of this lack of biological activity. And then what's very interesting is when the rains finally did arrive, the neighboring property on the other side of the fence, the water just sits on that surface. It's totally water repellent, and yet on this side of the fence, we see water sinking immediately into these soils. These are the soils that we've created around the planet that are either flash flood and drought, flash flood and drought. And what's key to this is microbiology and stimulating active fungi in the process. So there's a whole lot of reasons we should love fungi. One part of that is that they are forming these large soil macro aggregates. So think of soils really as like an apartment building, and who builds the very first bricks is bacteria, right? They're holding that brick together. Who provides the mortar to build the walls in your apartment building is the fungi, right? And so if we look underground, we have a whole metropolis of microbiology, and under that ground we have hallways and corridors and bathrooms and surfaces like banks and hospitals and schools and even a pub, right? And the microbiology are moving around this metropolis, and it enables them to have services that they're then providing to the plant. If we are in a more bacterial environment, we lose those services, all right? That soil starts to compact, we lose the walls and the hallways, those apartment structures, and then soil starts to move or blow. So it's absolutely critical that we have fungi building this structure in that soil, because that's how we get air and water movement. It's also key to reducing weeds and diseases in any of our ecosystems, is that these guys are so critical for decomposition and suppressing disease. So fungi, on the other hand, they feed on more complex foods. So bacteria might be feeding on the simple sugars, so green grass, your fungicides, your anything to kind of like sugar is a bacterial food, whereas fungi require more complex carbons, right? So they're what we call the brown materials. So those of you that have composted before, you know what I mean when we talk about browns and greens. So the green material gets hot. That's the activity of biology, of bacteria respiring, metabolizing, and reproducing in simple terms. And that's what makes that heat. Whereas fungi like the brown, they like the brown leaves, the cellulose, the lignin, your wood chips. Some of the things that we might feed them might be like straw or stubble, or I really like fish hydrolysate, your humic acids, biochar, wood chips, compost. All these materials become a resource for fungi to feed on. So those fungi are what we call our saprophytic fungi. We also have what we call our mycorrhizal fungi. So mycorrhizae actually inhabit on and inside plant roots, right? And it's a vital way that plants are actually accessing phosphorus, zinc, nitrogen, and other essential elements for plant health. It's also how plants can defend themselves, so these will wrap around their plant roots, and it's what is enabling those plants to reduce drought susceptibility. Mycorrhizal hyphae, so these fungi live in and on plant roots, will expand far deeper through that soil profile, and they're much, much finer than a root hair or a root in itself, and so it can access water that the plant itself cannot reach. So if you can imagine, on the left-hand side, this is a plant root without the presence of mycorrhizae. That plant may be reaching perhaps 2% of the soil volume for nutrients and water, whereas with mycorrhizae, so AMF stands for arbuscular mycorrhizal fungi, those plants can access potentially 20% of that soil volume. So you can see how much more nutrient and volume they're able to reach into. It might seem obvious, but the planet is driven by sunlight capture. And our performance, our profitability, our success, health, everything starts with our ability to capture sunlight. So it's that process whereby a plant breathes in carbon dioxide and then breathes out oxygen that enables us to live on the planet. But what's really interesting is part of this process is the plant takes in that carbon dioxide and it converts that through the energy of the sun with different types of protons that are hitting that leaf. They split that atom and they turn that into carbohydrates and then breathe out oxygen. So it's the carbohydrate we're really interested in. So carbohydrates consist of carbon, hydrogen, and oxygen, right? The plant will use that to turn that into plant materials. It's going to grow roots with that. But then potentially it may be sending 30% of those sugars that it's accumulating out its root zone to stimulate microbiology. So for us to be able to use some simple tools such as a refractometer, we can actually assess how well is this plant photosynthesizing. It's a very simple tool. It measures sugars and the dissolved solids. So for optimal grass health, we should see a Brix or percentage sugar above 12 degrees, right? So you look through that lens and take a look and see how well is this plant photosynthesizing. The blurrier the line, the better. So if we see very sharp lines, typically 3 and below, when you look through that lens, it's telling you that you have nitrates in the system. Why that matters is that that could be causing either insect, frost, or pollution issues, right? So get really interested in how well is my plant photosynthesizing right now. This is really important if we're thinking about carbon drawdown and greenhouse gases, which are increasingly coming under scrutiny. So we have two cycles that are going on, two main cycles in terms of carbon. So we have that short cycle, which is that grass clippings, compost, manure, whatever might be going on that soil surface. The plant and the microbiology might be breaking that down. So the microbes are chewing away on those different organic compounds, and then they breathe that out. So there's a breath in and a breath out every day with microbiology. Right, so it's an important cycle. It's just a short-term cycle. I talk about it like I'm here for a good time, not a long time honey cycle, right? So we see this process happening every single day, and it's very important. And it's why the stomata on your leaves or in the underside is they actually want to breathe that carbon dioxide off the soil surface, right? It's more efficient for them. That carbon is providing a home for microbes. It's also improving our water holding capacity and reducing that variability and stress on plants, right? So critical that we're building these carbon layers at not only shallowly in terms of organic materials and dead roots and dead worms or whatever is breaking down in here. We don't want to see any thatch, but also what we see is that deep drawdown of what Dr. Christine Jones calls the liquid carbon pathway, but basically think of it as liquid nectar that plant is pumping down into the root system to feed microbiology. That mycorrhizal fungi will take it far deeper down through that soil profile, and now we're really sequestering carbon. So in this picture, you can see this is a conifer plant that doesn't have mycorrhizae present. I want you to keep in mind that mycorrhizae in grasses is invisible, but in conifers we can see they have a slightly different variety. So you see they don't access much of that soil, but with mycorrhizae, suddenly that whole system comes alive. We have all of that access to nutrients and water, and basically what the plant does is it takes those byproducts from photosynthesis and uses that to pay for services. So it's a little bit like you've ordered pizza for takeaways or delivery, and so you send out a message and you pay for that with money, but the plant pays for it with carbon. So the plant is basically saying, hey, I feel like pizza tonight, sends out a signal, and in comes that pizza, or in this case it might be water or phosphorus or zinc. Right, so that microbe is responding to the signal from the plant about what the plant needs. The plant pays for it using carbon or sugars, and in return gets these benefits. So we really need to ensure that we do have adequate mycorrhizal fungi in turf or park systems. There have been some studies to show that we see improved turf coverage, we get root and shoot growth, we see an improvement in clipping yield or biomass, we start to see our water use efficiencies improve, and we certainly see faster grass establishment. There's lots of different ways we can increase our mycorrhizal fungi. First of all, we should stop killing them. So how do we kill them? Well, we kill them by high amounts of soluble phosphorus. So when you apply soluble phosphate, it actually sends a signal. The plant says, you know what, I'm not paying for you anymore. I'm not interested in your services. I have more than enough phosphorus, and it'll cut off that connection. And the mycorrhizae cannot survive without that connection to a plant. We also want to make sure we're avoiding pesticides and fungicides because they too will shed this relationship. They've shown things like glyphosate will actually decrease every following year that mycorrhizal colonization with the plant root by about 23%. So we're on a slow decline of this colonization or the health of mycorrhizae. We also want to make sure we're optimizing water. I'm sure you're not waterlogging your systems, but if you do, that's something to look at for why you might not have mycorrhizae. So instead of using soluble phosphate forms, we could use more microbial friendly forms of phosphate. So that might be guano or rock phosphate or fish hydrolysate. We can also feed soils with complex carbon forms. So these humic compounds, compost that are going to really help stimulate the mycorrhizae. Anytime we're disturbing, so that might be you have applied a fungicide or you have applied soluble fertilizers, you're going to come out and look at how do I repair that disturbance event by adding some kind of carbon that's going to stimulate my beneficial fungi. You could also looking at providing refuge species, so alley crops, pollinator strips, wild areas that are actually helping to recolonize and send spores out through that soil environment. So that is absolutely critical that we start to look at that.